Hello, science geeks. Welcome to Late Night News, and I am your favorite host, Areeb Siddiqui. Today on Late Night News, we have an interview with the revolutionary man, the man that can change the world, the one and only Dr. Masahito Tachibana. He is the man that has revolutionized the world of cloning through his discoveries in the field of embryonic stem cells. You better believe it, that someday in the distant future, there will be a clone of you sitting in front of you. Anyways, back to the interview. Mr. Tachibana, it is a great honor to have you with us.
The pleasure is all mine.
So, Mr. Tachibana, I have heard that you have made a massive accomplishment in the world of embryonic stem cells. Would you mind giving us some insight into your research?
Well, first, let me start with basics. Embryonic stem cell research has been going on for years, and I guess we were the first ones to find a real breakthrough. Pluripotent stem cells possess the ability to divide for long periods and retain their ability to make all cell types within the organism. The best known type is an embryonic stem cell. Our mission was to somehow get to stem cells by producing embryos, since stem cells are found in the development stages of an embryo. Now the thing with stem cells is that stem cells from a different body may not work in another body. So we need stem cells that are genetically identical to the body. In order to do this, we have a process called somatic cell nucleotransfer, in which the nucleus of a patient's somatic cell is transferred to an MTA, meaning an egg without a nucleus. Then the embryo starts developing with the genetic information of the patient and starts producing stem cells that belong to the patient. Although this technique was working perfectly with animal eggs, it was not working with human eggs. After installing the nucleus in the embryo started dividing, but it did not go past the 6 to 12 cell stage. And this is where we came in. We realized that the reason somatic cell nucleotransfer was not working with human eggs was that the oocytes were premature and not fully developed. The oocytes are premature eggs. So basically, in the past, we were experimenting with premature eggs, which resulted in a failure. But when we realized this problem, we started to use mature eggs, and due to this, we developed the embryo to the blastocyst stage, which is around 150 stem cells, enough to produce stem cells. So we basically made a fertilized egg grow to the point that there were stem cells.
How is this vital for the future?
Well, if we can find a source of stem cells, the world of medical science could take a major leap. Imagine if someone needed some brain cells. Brain cells cannot reproduce. So if you have 100 brain cells, you will never exceed 100 brain cells. But brain cells sometimes die out, and there is no way to reproduce them. But through the use of stem cells, we could potentially turn them into a nerve cell or any other type of cell that could help in healing or rehabilitation. If someone was to suffer from heart disease, we could replace the dead or decaying heart cells to new heart cells formed from embryonic stem cells.
What are some previous benefits we have seen from stem cells? I've heard that it has helped people's sight.
This is why stem cells are very, very vital for the medical field. Stem cells have been used previously to treat eyesight loss. Stargardt's macular dystrophy is an incurable disease developed in childhood which causes gradual loss of sight. UK medics have conducted a surgery on 12 patients by injecting retinal cells that were formed from stem cells into the patient's eyes. If such use of stem cells can be used to treat a variety of diseases, it can be a revolution in the medical world, and this is what we're looking for.
What alternatives do you have for embryos that will please critics?
Many critics think that since cloning animals is dangerous, then cloning humans is also dangerous. So they've come with a new way to create stem cells. The technique uses skin cells and converts them to induced pluripotent stem cells through the use of proteins. Induced meaning persuasive or believable stem cells. Although this method is relatively cheap compared to embryonic stem cells, there are still a lot of questions about the quality of the stem cells produced.
Are there safety concerns with stem cells?
It is believed by some that stem cells could pass viruses and other diseases to the patient when they get a transplant. Some also believe that stem cells could turn into cancer cells. Research has shown in animals that the use of stem cells can cause malformed body parts. This could be one of the down effects of cloning because it can be dangerous. But at the moment, we have just started to explore the world of embryonic stem cells.
This is it for tonight, folks. Thank you, Mr. Tachibana, and see you next time on Late Night News.